Good, man. How are you? Good to see you.
Nice to
Thank you. I was just trying to figure out how to command this. Okay, I'll do it with my laptop and it'll appear on here because I'm sharing my screen.
Cool.
Cool. I think we got it.
Let me, I'll do sort of announcements and I'll
Sounds
introduce.
yeah, sounds good.
It's you, sorry, you, Sarah, Josh.
Yeah.
Cool.
Cool.
All right. Hey, good morning. A couple of announcements just to start our morning for geriatric grand rounds. Next grand rounds is going to be October 16th, 2025 with Lauren Abate, and she's going to be talking about mobility in the geriatric ED setting. And our next journal club is going to be on November 13th, 2025 with Gretchen and Kate. Other announcements include that we have a division meeting after today's presentation. Everybody will get a link with recording of today's presentation and some PowerPoints after today's grand rounds. And in the email, you'll also get an online evaluation, or you can complete yours in person with the paper that Jessica is going to hand out in the middle of the presentation. Submit questions in the chat. We'll try to facilitate and make sure that they get answered if they do appear in the chat. And without further ado, I'm very excited to talk about today's geriatric grand rounds. At our prior division strategic meetings, we were talking about how sometimes with research, we do get small findings that come out. And as a division, we're really working to try and get a better understanding of what everybody does on a regular basis. So rather than, wait until someone's got a full hour of stuff to present, we talked about how we might be able to get our researchers to present little mini updates in this lightning rounds format. So it's a little bit, I guess, like the AGS stuff, but sometimes it's not necessarily for a conference. We're not trying to make it that formal. We just want to create a structure where we hear a little bit more about what we're doing and invite people to sort of think about that moving forward. So very bravely, Zach, Sarah, and Josh agreed to present small updates from their research, and I'm very excited to hear what they have to say. Just a little bit about format. They're going to do 18 minutes for each presentation, and after each presentation, just so that we can, again, talk about these ideas as a division, they'll open up the floor to comments and they can handle it, but I'll facilitate handing the mic around. All right, without further ado, Zach, Clayton.
Thank you everyone for being here. Thank you, Kenny, for the introduction. We're excited to engage in this style of format, so look forward to some discussion. So I titled this Breast Cancer and Premature Vascular Aging, Current Understanding and Putative Therapeutic Strategies. So there will be a little bit more outside of this as what other projects we're working on in the cancer space and premature aging. But for the most part, we'll be focused on breast cancer. All right, so a little outline for today. We're going to talk about cardiovascular diseases, and it'll be abbreviated as CVD for short, cancer and aging. Then we'll go into our ongoing preclinical work studying breast cancer and the various model systems we use to engage in that process. Our ongoing clinical work studying lymphoma survivors. Next steps in our ongoing work. And then lastly, I hope this will really spark some discussion and thinking about ways in which I think we can engage geriatricians in our lines of investigation. So we're going to start out with some broad public health statistics, and I don't think any of this is going to be too surprising to anybody in the room. But just to reemphasize the point, here's some data from the American Heart Association demonstrating that cancer and cardiovascular diseases have been, remain, and continue to be the primary causes of death in the United States and are projected to remain that way for decades to come. But cardiovascular disease and cancer risk are also both a product of aging. So although we often talk about in our work that aging is the primary non-modifiable risk factor for the development of cardiovascular diseases, but it's also the case for cancer. So take another data set from the American Heart Association and you plot decades of age on the x-axis and the percent of the population with cardiovascular diseases. You see that the increased prevalence of cardiovascular disease is primarily a product of advancing age, such that 95% of cardiovascular diseases occur in middle-aged and older adults. But also similar trends persist when we look at data from the Social Security Administration and we look at cancer prevalence relative to to advancing age. So just to reemphasize the point, aging is the common linchpin between increased cardiovascular disease burden and cancer prevalence. But we've become increasingly interested in the crosstalk between cardiovascular diseases and cancer. And some of you likely know that there's a field of cardio-oncology that blends the concepts of cancer care, and cardiology. And there's cardiologists that end up specializing in cardio-oncology. So I'm just going to show some data from a variety of large population cohorts that have been published just this year in what we consider the reference standard journal for the cardio-oncology field, which is the Journal of the American College of Cardiology and their cardio-oncology sister journal. So this is well known that cancer and its associated therapies drive cardiovascular disease risk. But here I just wanted to use an example to demonstrate how various large population cohorts and data sets have been used to help ascertain the connection between cancer and cardiovascular disease. So these group of investigators engaged with a Danish population-based registry, and they studied over 550,000 individuals to demonstrate this connection between cardiovascular disease risk in cancer survivors. And as I mentioned, this is where the field of cardio-oncology has really emerged, the influence of cancer and its associated therapies on cardiovascular disease risk. But I think what's becoming increasingly more appreciated these days and over the last few years is this concept that cardiovascular disease is actually a primary risk factor for cancer. So a group out of Stanford engaged with some market scan data, and they studied over 130 million individuals, and they found that cancer risk was markedly increased after the diagnosis of cardiovascular diseases. So now we have this connection between cardiovascular disease driving cancer, and the field has broadly characterized this as reverse cardio-oncology. So now to blend the two concepts, we have this unfortunate vicious cycle that if we have our older adult that gets diagnosed with cancer, they'll often go through chemotherapy, and that will drive cardiovascular disease risk. But now we know that from emerging data from that study I just showed and others, that cardiovascular disease is actually a driver of cancer. So when we look at this vicious cycle, we ask ourselves where might be, especially as cardiovascular physiologists, where might be the best place to intervene? And we believe that's at the level of cardiovascular diseases. So if we can ultimately reduce cardiovascular disease burden, we may be able to reduce cancer burden and then subsequently put a halt or a break on this vicious cycle. So to begin addressing this problem, we ask ourselves, why is aging a primary non-modifiable risk factor for the development of cardiovascular diseases? And one of the key drivers of cardiovascular aging is a process called vascular dysfunction, which I'll refer to herein as vascular aging. And when we talk about vascular aging, what we're primarily referring to is stiffening of the large elastic arteries. That's primarily the carotid arteries and the aorta. Most of our work focuses on the stiffness of the aorta and endothelial dysfunction, so the inability of a vessel to dilate to the degree that it did when that individual was young or that animal was young. So I presented this material last December in Grand Rounds, so I'm just going to give a general overview of how we study these two main physiological outcomes in the laboratory across the translational continuum from human to mouse. In humans, we perform a measurement, a bioassay called carotid to femoral pulse wave velocity using applanation tonometry, ultimately just gathering arterial waveforms from different arteries and looking at the time delay that it takes from blood to travel from one artery bed to the next. And we can ultimately measure the distance between those two sites to calculate a velocity. And we can also leverage that same principle in mice in vivo while mice are under anesthesia. We can assay the aortic arch and the abdominal aorta and essentially do the same thing. And importantly, we try to only make measures in mice that, especially on the physiological level, that are clinically relevant to humans because we want to do mouse work that ultimately informs human health. But as you can appreciate, a mouse model does allow for the ability to excise arteries, a tissue not readily available in humans. So what we can do as well to complement our pulse wave velocity measure in mice is dissect out aortas, and we cut the thoracic region of the aorta, which is the primary pathophysiological region that stiffens with advancing age. We cut it into approximately 1 millimeter segments, add it to a pin myography system, and perform incremental stress strain testing, which allows us to leverage an engineering principle termed the Young's modulus of bend and essentially calculate material stiffness of the aorta. So now we can look at strictly aortic stiffness independent of any complementary effects on the stiffness of the artery in vivo, such as blood pressure, neuronal input, or humoral factors. In the context of endothelial function, we study this clinically through the reference standard measure called brachial artery flow-mediated dilation, where we leverage a mechanical stimulus to drive nitric oxide production, a primary vasodilatory molecule that's released when there is increased blood flow on the vessel wall, termed shear stress. And we can look at how the diameter of that artery before and after that blood flow stimulus. And we drive that blood flow stimulus by occluding blood flow just distal to the elbow with a blood pressure cuff. And our complementary bioassay in mice is we excise arteries from mice, and we primarily use the carotid artery, but we have work going on in the laboratory from our group and others where we're looking at muscle feed arterioles, brain blood vessels, et cetera. And we can also do this in rats, which is complementary to another project going on in the division with the SCORE project. And so just to leverage this bioassay, we can take a variety of approaches to understand and dissect out the mechanisms that might underlie differences in endothelial function. And here's just this is an artery that Brandon Wynn cannulated a while back. And so what this is a mouse carotid artery you tied to stainless steel cannula, and we can use also glass tip cannula, and they use fine microsutures. So what this is a training microscope that allows us to visualize what we see under the scope. And so for those that are learning how to do this process, this additional iPad-like feature on top of the microscope serves to be beneficial for training purposes. So that's an overview of our clinically relevant measures. And I bring these up because I will show data related to these outcomes later on in the talk. So with that as background, we'll now get into our ongoing preclinical work studying breast cancer. And for some context, most of my postdoctoral work did not use cancer-bearing models. So that's been a new, exciting line of investigation to pursue upon coming to the Anschutz Medical Campus and becoming a member of the Colorado Cancer Center. So I imagine many people are aware of the alarming statistics related to breast cancer, but just to remind you, unfortunately, 1 in 8 women will be diagnosed with breast cancer in their lifetime. But what is known is that breast cancer is very prevalent in women, but what's becoming increasingly appreciated is the cardiovascular disease burden that breast cancer survivors face. It's a little bit of a busy slide, so I'll walk you through it. But this paper published in 2011 titled Cardiovascular Disease Competes with Breast Cancer as the Leading Cause of Death for Older Females. So here we have different stages of breast cancer, stage 1, 2, and 3, or 3, 4 blended at the end. Cardiovascular disease as a cause of death is the solid white bars. Breast cancer as the cause of death is the solid black bars. And across these different age ranges, you can see at stage 1 and stage 2 of breast cancer, cardiovascular disease remains a large cause of death. And then upon these later stages, the breast cancer perhaps is too advanced for them to live past that stage. And this article goes on to discuss that it's likely because the treatment regimens we have to target breast cancer become so effective that patients are unfortunately living into their later years and dying prematurely of cardiovascular disease. And indeed, breast cancer survivors are at 2 to 3 times higher risk of developing cardiovascular disease relative to age-matched women. So our preclinical studies of studying breast cancer, so I wanted to just give a general overview of the 2 most common approaches to study cancer models. And in case you're unaware, these are things that we've learned over the last year and a half or so. And one common way is a PDX model. So that's a patient-derived xenograft. So that's where we administer actual human cancer tissue into mice. But given the challenges in immune reactions and immunoglobulin incompatibility, we often have to use immunodeficient mice to administer the human cancer line. Or you can use a syngeneic model, which just most simply is defined as immune compatibility. So you can take a mouse cancer line and inject that into a mouse. And we have ongoing lines of work using both model systems. So now to get in our PDX model, when I first arrived here and then Brandon joined me as a postdoc last September, we got to know Dr. Peter Cabos over in Med Onc, and he had a ongoing line of investigation where he was essentially explanting human breast cancer cells into immunodeficient mice, and they were interested in also a dietary intervention termed intermittent fasting, and they have that going in mice and in humans. But there's been mixed literature as to the effect of intermittent fasting on the vascular aging process. When we didn't quite have a lot of material set up, but what we knew is we had drive, time, and some dissection tools. So we went over to Peter's lab over in RC1 North and dissected all the aortas from his animals and saved them in a way where we could try to create a story and determine if there's a premature vascular aging phenotype in his animals. And importantly, these are in young female mice. So like I said, he gave a luminal B human estrogen receptor positive breast cancer cell line to immunodeficient mice and had them undergo ad libitum feeding or intermittent fasting. And that led us to have 4 groups. So control plus ad libitum feeding, control plus intermittent fasting, our 2 breast cancer models with ad libitum feeding or intermittent fasting. So this allowed us to look at, in the context of ad libitum feeding, the direct effect of breast cancer on the vascular aging process and perhaps the mitigating effects of intermittent fasting. And like I said, we didn't quite have all of our primary physiological equipment set up at the point in which we collected these tissues, but we were able to, because we were able to get the aortas from his animals, leverage our ex vivo assay of aortic stiffness. So that's where we perform incremental stress strain testing. And so what we found in the ad libitum fed group, cancer-bearing animals are in the red, that they had higher stiffness, so as measured by aortic elastic modulus, and that the breast cancer group that had undergone intermittent fasting were largely protected from this phenotype. And surprisingly, no one has really tried to characterize the independent effects of cancer on the vasculature. A lot of the work to date has been focused on chemotherapy. So this is showing us that perhaps just breast cancer alone could be driving a premature vascular aging phenotype. And not showing you all the data that we have thus far, but 2 other characteristics of vascular aging that occur in the aorta, one of which is the degradation of elastin. So elastin is the protein in our arterial wall that helps help keep the vessel elastic, but it does degrade with advancing age. And we can actually calculate elastin fragments in aorta cross-section using histological approaches. And complementary to our aortic stiffness data, we found that there was elevated levels of elastin fragmentation, so that elastin is becoming less functional, perhaps, and that's what you can infer from these data with the effect of cancer. And this was largely mitigated in the cancer setting with intermittent fasting. And we actually found markedly thinner aortic walls. So could perhaps influence the permeability of the blood vessel and allow immune cells, for example, to more infiltrate into the vascular wall, and a variety of other pathological factors. So we found in the setting of cancer, we actually had some markedly higher levels of wall thinning, which seemed to be protected with intermittent fasting. And I honestly, I cannot do all this work by myself, so I have to give a big shout out to Brandon Wynn and Ravi, our coordinator and senior clinical sciences professional, for driving a lot of this work forward and working with the variety of individuals that we've had the fortunate opportunity to have come in and out of the lab over the last 6 to 8 months. So now into our syngeneic models. Again, I'm just trying to give an overview of what we're doing in the lab and not take any big deep dive on any of these topics. Hopefully for future grand rounds, we can take a deeper dive on these. But for our syngeneic model study, we were fortunate about last November or so, we got a pilot grant from the Colorado Cancer Center through their molecular and cellular oncology subdivision to understand how breast cancer and its common treatments influence the vascular aging process. So here again, we used a breast cancer bearing animal, but this time we engaged with a mouse breast cancer line, which allows us to use normal healthy mice rather than immunodeficient animals. And this is specifically mice bearing E0771 cells, which is a common mouse breast cancer line. And so rather than doing a dietary intervention in this case, we were interested in studying the influence of common breast cancer chemotherapy regimens. And we went with adriamycin, which is also another name for doxorubicin, plus cyclophosphamide as a common cocktail that's used to treat breast cancer. So it gave us 4 groups of a healthy control, chemotherapy alone, breast cancer alone, and breast cancer plus chemotherapy. And by the time we euthanized these animals, we did have our all of our equipment set up, so we were able to engage with our endothelial function and in vivo aortic stiffness measures. And again, this work was largely driven and led by Dr. Brandon Nguyen and Ravi in the lab. So here we leveraged our carotid artery bioassay where we cannulate them to the stainless steel pipettes that I mentioned earlier, and we perform increased dose. We expose the vessel to increased doses of acetylcholine, which is a vasodilatory molecule. And what we know is if we compare young versus older mice, that we see old mice have a relatively similar magnitude of reduction in endothelial function as we're seeing with chemotherapy, cancer only, and cancer plus chemotherapy. And right before euthanasia in a subset of animals, this was another piece of equipment that we didn't have set up yet quite in line with this. We were able to do this in all of our animals. But thus far, we do have some exciting data, we believe, showing that there's a relatively similar level of aortic stiffening as measured by aortic pulse wave velocity in our chemotherapy-bearing animals, cancer-bearing animals, and then the combination of the two. And we had hypothesized that the cancer group that had also received chemotherapy would actually have worse vascular function than the other groups, but we're not actually seeing that. So we're performing we have a lot of other complementary data to what I'm showing you here, and we're still engaging in this process, and I'll give you a snapshot of what that looks like here in a minute. But nevertheless, this is an exciting line of investigation that we're excited to continue to pursue. So our ongoing clinical work studying lymphoma survivors, I briefly touched on this in my last grand rounds, but this study is off and running now, large thanks to Ravi in the lab. And then this study is funded by the NIH through an R00 mechanism. And I was fortunate to get additional funding to support additional measures through the CCTSI CTRC microgrant program. We're titling this study the SENDOC study, so you might see some flyers all across all the elevators where we're trying to recruit adult lymphoma survivors and age and sex match controls. And we're calling this the SENDOC study as to study the cellular senescence, doxorubicin chemotherapy, and cardiovascular health. And we ultimately chose lymphoma survivors because they're often treated with doxorubicin chemotherapy. And for this study, I was fortunate to create collaborations with Dr. Lavanya Kandapalli, who directs the cardio-oncology program in the Division of Cardiology, and as well as Dr. Manali Kamdar, who runs the lymphoma services. And upon patients finishing treatment with Dr. Kamdar, she forwards us their information, and then we're able to engage. So that's another pipeline of recruitment for us. And again, a big shout out to Ravi for really driving this project forward and navigating all the hurdles it takes to do a study like this on this campus. So the outcomes we have in this study are endothelial function, arterial stiffness as measured by aortic arterial stiffness. We're also looking at stiffness of the carotid artery and the intima media thickening of the carotid artery. We're looking at cardiac function by cardiac echocardiography. We're looking at cerebrovascular function and cognitive function. We're looking at hand grip strength and body composition by DEXA. And we're performing intravenous endothelial cell biopsies to try to get a more integrative look at what might be changing in the vasculature at the cellular level that could be complementing or being related to what we're measuring in vivo. We're also taking a variety of blood samples, serum, plasma, to look at a variety of humoral factors. And we're engaging patients with a lot of different questionnaires, such as general health survey, social determinants of health, sleep quality, mental health, habitual physical activity, habitual diet, gynecological history, and in the case of our cancer survivors, their treatment history. Because all of these factors have shown to influence the vascular aging process, and we are interested in how these factors might we want to consider these factors as covariates in our subsequent analyses and determine how they might be influencing the premature vascular aging effects of chemotherapy treatment in these survivors. And upon completion of the study, we'll do a variety of bioassays with the serum where we expose them to naive cells or blood vessels from mice to determine the influence of humoral factors on vascular function. And we are funded to perform metabolomics and proteomic analyses of those blood samples. And we're also isolating a variety of cell types from the blood to assay different molecular markers of cellular senescence. So the next steps in our ongoing work, in our PDX model, we have engaged with the Extracellular Matrix Characterization Center that's housed here on campus in the structural biology and biophysics core facilities. And we're performing extracellular matrix specific proteomics because if you recall, like our primary physiological readout was arterial stiffness, and a major molecular mechanism that drives elevated arterial stiffness with advancing age is adverse changes to the extracellular matrix. And historically, we've measured proteins one at a time, a particular collagen isoform, an elastin isoform, but this allows us to take a more shotgun approach and look at a variety of different isoforms. So we should have those data in hand in the next week or two, but it's providing you an example from one of that group's previous publications. So you can look at insoluble and soluble factors, intracellular components, and you can look at all these different collagen isoforms, transglutaminase isoforms, and a variety of other factors that might allow us to then engage these proteins as therapeutic targets in the future. So then another step and another aspect of our molecular and cellular pilot grant with our syngeneic model is to ask the question, does exercise prehabilitation by way of high intensity interval training mitigate the premature vascular aging effects of chemotherapy treatment? So to brief overview, the animals will bear cancer. They will engage in high intensity interval training. And we have some fun videos that have been edited with fun music that I'll show you at a different time with mice running on these treadmills. It's pretty neat to watch. And so they'll exercise up to the point of getting their chemotherapy to ask the question during that sensitive period from cancer diagnosis to the onset of chemotherapy, can we advise patients to undergo a lifestyle therapy that might help mitigate that stimulus that drives premature vascular aging? Then after collecting the vessels from these animals, we'll combine them with our sedentary data that we have thus far and perform RNA sequencing. And just shout out to Brandon and Ravi and Dr. Cabos as we just published this review, kind of overviewing this concept in exercise and sports science reviews, one of ACSM's journals. And I thank Carrie Moreau for the opportunity to write that article. Next steps in our ongoing work. I think we're excited about this line of work is we're now asking the question, how does the compounding effects of advanced age and chemotherapy treatment influence both vascular health and brain aging? So the general research question or aim is to determine the degree to which advanced age influences premature vascular and brain aging following doxorubicin chemotherapy treatment. And this is hinging on some work that I was fortunate to do during my postdoc, where we demonstrated that doxorubicin drives a state of premature vascular aging in young mice. And at least at the level of the hippocampus, we found that the hippocampal transcriptome of young animals that were given doxorubicin had a transcriptome that was very similar to old mice and older and cadaver hippocampi. So this work is going to be largely driven by Doctors Brandon Wynn and Mackenzie Kmeyer. And Mackenzie's bringing a unique skill set to where we can complement this hippocampal transcriptome data and actually look at cerebrovascular function. And I also wanted to, knowing that we all engage with the National Institute on Aging, emphasize a very, what I would just call a cool feature of the Institute. So the National Institute on Aging has a division of aging biology. And within that division, there's a translational research branch that houses an aged rodent colony. And for perspective, if you have an NIA-funded grant that is proposing to study old animals, you get 20 free mice per month. But if you want to buy these animals, they are on the upwards of $300 to $350 per mouse. The NIA was blasting out a lot of emails saying that they have a lot of leftover mice to study. So we were fortunate to acquire over 100 animals for free from this colony. So we have young and middle-aged animals. And so now we just have to pay the per diem and aren't having to pay that 300 plus per mouse cost. So I think Brandon and Mackenzie and Carrie we all met to organize this effort, and we're really excited to move this forward and look at the influence of move our cancer and chemotherapy work more into the true context of aging. And then lastly, we'll end with ways in which I think we can engage geriatricians. As been stated before, Kenny and I have had the fortunate opportunity to try to work together to try to figure out how we can leverage databases. But as I continue to think about this, I was wondering if we can assess aging relevant trends in cancer survivors. such that is there something that we can look at that from large data sets that help us better understand is are cancer survivors experiencing a state of accelerated or premature aging? Can we determine if baseline aging characteristics influence the premature slash accelerated aging effects of cancer treatment? And then in our ongoing clinical study, are there aging relevant questionnaires we can be administering? We want to think beyond what we're already doing, and would very much appreciate any additional perspectives. When we are interacting with middle-aged and older adults, again, we're recruiting patients 18 and older. Are there certain things we should consider from the perspective of bedside manner? We want to be sensitive to that always. And then how might we best, something I've been particularly interested in, like I said, we are quantifying mental health via a questionnaire in the patient, but from the perspective of those of you that interact with caretakers from the lens of Alzheimer's disease and related dementias, it got me thinking, how might we best quantify the mental health burden of the caretaker and how that could influence our study outcomes, such that if the caretaker is experiencing an elevated burden of mental health, could that potentially influence our outcomes and then subsequently influence the mental health of the patient? Um, with that, I just want to say thank you again for the opportunity to speak to you all today. And I, as any PI, can't do this without the help of their team and the great staff. So thank you.
I think we'll take time for one question just because we want to leave time for others, but any questions from the audience? Or any responses to these questions, actually, room full of geriatricians.
We can talk later.
Carrie?
With your last question, I know we've thought about this caretaker burden in the context of PTSD because a lot of the caretakers can also develop PTSD. And it might be interesting rather than thinking about how they're affecting the care of the individual, but bringing both in to see, you know, how those environmental stressors that both are being exposed to could be affecting their vascular aging as well.
Thank you.
Next up, we have Dr. Sarah Wherry.
I don't have to ask you if you can see this. I can actually verify that you can see it. All right. So I'm going to be talking specifically about one of my studies that I had done related to bone biomarker responses. So I'd given a grand rounds on this, I think about 2 years ago now, and I had talked about this experiment. So now here's at least the update of what has happened there. So starting out with a little bit of a background for those who haven't heard me talk about this before of the relationship between bone and exercise. So generally speaking, we think that exercise is good for the skeleton. So bone responds to mechanical load. It's a mechanosensitive tissue. So whatever load you place on it, it should adapt to. And exercise increases mechanical loads through multiple types of forces like tension, compression, shear, and torsion forces. And from epidemiological data, we do know that people who are regularly exercising have decreased non-traumatic fracture, osteoporotic fracture rates than people who do not regularly participate in exercise. But there is a little bit of a caveat here that this is at least partially due to some indirect effects as well, not necessarily bone effects. So exercise has a lot of benefits. It's going to improve balance. It's going to improve muscular strength and endurance. And all of those things are going to reduce fall risk. So if you're not falling, you're likely not fracturing your hip. So maybe some benefit on the bone, but largely also contributing to these other types of effects. And certain forms of exercise are going to be considered more beneficial. So a weight-bearing exercise, such as walking or running, is generally thought to be more beneficial to bone than a weight-supported exercise like cycling or swimming, although both of these types of exercises are going to exert forces on the bone. The benefit of the weight bearing is you also then have to deal with gravity and supporting your own body weight. So this picture here, which I think many of you have seen before, is just capturing what I just told you. So we're looking at two different types of athletes here. We have runners versus cyclists. And not surprisingly, runners have higher bone mineral density across these various clinical sites compared to a weight-supported athlete, or sorry, the weight-bearing athlete of the runners. But what's interesting is there's something about exercise that seems to be able to induce bone loss under certain conditions. So this is a group of young male cyclists followed over a year of training and competition and their off season. And in that 9 months of training and competition, they lose about 1.8% of their hip bone mineral density. In the off season, they get that little bit of recovery. So they get up to about 1.5% loss overall across 12 months. But in that time of recovery, they never get back up to they were at the beginning of that 12 months. So as you imagine, if someone's participating in these types of sports continuously over many different seasons, could be increasing their risk for bone loss and their risk for later fracture. Seeing the same thing in female cyclists over a same time point, and to put this in context, this degree of bone loss, which is again younger adults, is similar to the annualized bone loss that you see during the menopause transition in older women. So a very significant degree of bone loss occurring in a population that really should not be having it. And there are a lot of reasons why this could be happening: energy disruptions, nutrient problems, sex hormone problems, stress. But we've chosen to focus on this idea of a disruption in calcium homeostasis. So we'll do a very quick refresher of parathyroid physiology so that as I get into my data, you all know what I'm talking about. So the calcium sensing receptor of the parathyroid is going to detect changes in serum ionized calcium. As serum ionized calcium decreases, there'll be an increase in parathyroid hormone, or PTH, which is then going to signal to defend that serum ionized calcium concentration. And there are three main ways that that can happen. So you can have calcium reabsorption from the gut, you can have calcium reabsorption from the kidney, but both of those require that a sufficient amount of calcium is available to increase that ionized calcium concentration. The skeleton is the largest calcium storage site in the body. So when you have a large need for calcium, you're going to start breaking down your bone in the short term to release that calcium into the bloodstream to get those concentrations back up. And this is going to feed back into itself until that serum ionized calcium concentration is stabilized. So we think about this in the context of exercise. We hypothesized this before, but we've now shown, at least through several studies, both us and some other labs, that exercise will cause a decrease in serum ionized calcium. That will signal for an increase in parathyroid hormone, which will then signal for an increase in bone resorption that we measure through this marker CTX, and again, feeds back into itself until that serum ionized calcium concentration is stabilized. I can tell you, as long as you are exercising, at least in our hands, that ionized calcium concentration never stabilizes. So you have active bone resorption going on the entire time that you're exercising, and you don't see that recovery until the exercise is ended. So a lot of this work has been done in endurance types of sports. And when we look at younger adults, this work has mostly been done during stationary cycling. In older adults, it's primarily been done in treadmill walking, and there's been a few younger adult running exercises. So just walking you through this figure here, the red bars are cycling, the blue bars are walking or running, the lighter shades are at a moderate intensity, and the darker shades are at a high intensity. And really all I want you to take from this is that it seems that cycling may be a little bit worse than treadmill walking in terms of the increase in parathyroid hormone, but then also increase in CTX, that marker of bone resorption that we tend to use. Also seems to be an impact of intensity, where higher intensity exercises seem to cause a greater increase in parathyroid hormone and bone resorption than a lower intensity exercise. It seems that older adults have a somewhat blunted response compared to younger adults at a similar intensity, but we're having a problem here of trying to be comparing modality to one to another, which was sort of the point of the experiment that I'm getting to, because the only treadmill exercise that was done in younger adults was running, whereas in older adults has been primarily treadmill walking. So the main piece of this was that it seems that there's a difference in this activation of bone resorption between treadmill walking and stationary cycling, but it hadn't ever been tested within the same individual. So we weren't sure if these differences between these modes of exercise were lab to lab differences, differences in the type of exercise performed, age, a number of things. So that led to this study, which was called EB for exercise and bone biomarkers. I'm not very creative at acronyms, but that's what we ended up with. So we had 9 older veterans. So being in the VA, primarily men, and they completed 2 60-minute exercise bouts that were separated by at least a week, and the order of exercise was random of which one they got first. So one was treadmill walking and one was stationary cycling at about 75% of their measured maximal heart rate. We drew blood every 15 minutes during exercise and then followed them for 4 hours after exercise to keep drawing blood. We discharged them and then they came back the next morning for another blood draw 24 hours after their last exercise bout. This is just a schematic of what the sampling paradigm looked like to get an idea of when we were drawing blood relative to exercise. And then I'll get into the data. So I'll walk you through these figures here just to help orient you. So on the top, so panels A and B, that is your serum ionized calcium. The one in panel B, where it has this little adjusted indicator here, that just means that these were plasma volume adjusted to deal with hemoconcentration that you get during exercise. And we do that because exercise-induced hemoconcentration can make some of your markers appear artificially elevated. So just an adjustment going on there. And then the bottom is the parathyroid hormone. So starting with the ionized calcium, we see a decrease in ionized calcium with exercise in both modalities. We've seen this many times before. This is not particularly surprising. This very precipitous drop that we see after we plasma volume adjust is also something we and others have seen multiple times. Parathyroid hormone, predictably, in response to that decrease in ionized calcium, will start increasing and will peak about 15 minutes after exercise is over, which is the same time that you have the lowest point in your ionized calcium. So seeing those nicely tracking together. And parathyroid hormone is not highly influenced when we plasma volume adjust, so you can see those patterns look pretty similar across the adjusted and unadjusted. But the other big piece of this is that there was no difference between these 2 modes of exercise. So looking at the cycling in the blue and the treadmill in the black open circles, when I first plotted this data, I thought I accidentally plotted the same data twice. So they effectively overlay each other. So this is suggesting, at least for ionized calcium and parathyroid hormone, that the mode of exercise did not matter when it's being performed at the similar intensity in the same people. Then going to our bone biomarkers, so CTX, that marker of bone resorption, and P1NP, our marker of bone formation. Similar thing where we have on the one side unadjusted and then adjusted on the other. Maybe there's something going on here and that first hour exercise is over between cycling and treadmill, but by the time you get out later into recovery, there isn't a difference between these two, and this difference was also not statistically significant. And that pattern holding when we plasma volume adjust. P1NP, again, maybe something going on at this 1 hour post-exercise time point that seemed to persist a little bit after plasma volume adjusting, but again, not statistically significant. And to put this in context, that degree of difference between those two is pretty small in terms of the markers overall. So then in conclusions for this, basically what I've already told you is when you're performing these modes of exercise in the same individuals at a similar intensity, the bone formation and resorption responses are very similar, and the ionized calcium and parathyroid hormone responses are nearly identical. So it does not at least see that mode of endurance exercise is an important contributor to this decrease in ionized calcium or parathyroid hormone. So the good news is, if you like a certain type of endurance exercise, it's not necessarily better or worse for you than the other. They're both equally bad, at least in terms of the short term for bone health. There are some future directions. So we really have not gotten at the effective age. So when we compare these types of things in younger versus older, there seems to be a greater response in younger adults. But younger adults tend to be able to exercise at a much higher absolute intensity than an older adult. So when you match them for relative exercise intensity, what does that mean in terms of absolute exercise intensity? So to give you an indication, in our studies of younger adults, they were cycling at an average of about 270 watts for the hour of exercise, which is a pretty high power output to sustain for that long. Our older adults with their treadmill walking are walking at about 3.2 miles per hour to 0% grade. And both of those are at 75% of their max heart rate. So matched from a relative standpoint, definitely not matched from an absolute exercise intensity standpoint. I'd mentioned before in that figure that there is a role of exercise intensity. So need to do a little bit more work of seeing how does exercise intensity also influence the response within the same individuals. And there is no apparent sex difference that we've seen so far. The men and women are responding very similarly, but we have also not been powered to necessarily detect that. There's still a little bit of work to be done in terms of exercise mode. So this study was looking really nicely at endurance types of exercises. We still don't know a lot about resistance exercise. So that is a study I have ongoing right now to look at, is there a similar type of pattern that emerges with resistance training? I can tell you from the very small number of preliminary data I have so far, it looks completely different. So there is no decrease in ionized calcium, no increase in parathyroid hormone, but this is an N of 4, so don't get too excited, that could change. And we don't know whether there is a training effect. So we've seen this same response in older, younger men, women, trained, untrained, weight supported, weight bearing. The pattern repeats itself, but we haven't actually tested it. So that's another study that's ongoing right now of doing exercise training and then measuring this at multiple time points across the training window to see if it changes as someone gets more accustomed to the type of exercise that they're doing. And then wrapping up here with some potential for collaboration. So I have a lot of broad research interests. This is very much the biomarker side of the work that I do, but as Elizabeth definitely knows, there is an imaging side of my work as well, of looking at bone mark architecture changes in response to exercise. So generally, I'm interested in how exercise affects bone health. So how can we prescribe exercise to improve bone mineral density and reduce fracture risks across the lifespan, but with a special interest in older adults? Curiosity about interactions between exercise and lifestyle behaviors in bone. So I have an arm of my work right now that's looking at weight loss and exercise and how that might be able to be used to preserve bone health, and how exercise might be able to be used in those at high risk for fracture. So with interest in things like improving falls or improving muscular strength and muscular endurance. So I just wanted to thank all the people who made this possible, especially Paige, who collected most of this data, and Christine Swanson, who is our study physician and co-investigator, as well as all of our funding sources. Thanks.
So apologies if I don't understand the graphs totally. What it seemed like was that the treadmill versus cycling was similar, but then we know that like weight bearing exercise is better for osteoporosis. So how do you hold this in the context of that? This is just the short term versus the long term.
Yes.
Why is it different?
So there's a bone remodeling response here in terms of calcium. So this is your body doing whatever's possible to make sure you can maintain serum ionized calcium concentrations so you don't go into cardiac arrest. So like, yes, you're going to need what you need to do to stay alive, but there's the mechanosensing piece of this as well, which these markers aren't going to be able to get at in the short term. So there's going to be some adaptation depending on the load. So the way we've sort of thought about this is there's a mechanical benefit of exercise, but there's also a metabolic response to exercise. So this is the metabolic response, and it may be that this, under certain circumstances, is undermining that mechanical response to exercise. So that too much of this, maybe then you're diminishing the benefit of loading the bone.
Hey, Sarah, nice work. Two things, an opportunity for you for collaboration if, given your interest in exercise, if you want to leverage my current study, which is studying adults across the lifespan and looking at exercise response variability, recognize we're not going to see DEXA changes, but I don't know if we could see the bone microarchitecture changes, but also we are getting blood samples before, during, and after exercise, and we could easily give you a blood sample or add on a blood sample if that would be beneficial, and we're taking an omic approach. So let me know if you're interested. And my question is, you say you don't know really if there are sex differences, but when you study it across the lifespan, you see the changes in hormonal status, or looking at it, both men and women who have normal eugonadal or hypogonadal states, could there then perhaps be sex differences?
Yeah, potentially. It's a lot of our work has been done primarily in men, unfortunately. So we've had small numbers of women participate, which has somewhat limited our ability. But when we plot men and women together and we separate them out by sex, they overlay each other, but we also don't have great information about gonadal status. So something we can do a better job collecting in the future. Thank you.
And last but not least, we have Josh Yarrow.
Thanks. Today I'm going to talk about testosterone replacement therapy, which I'm going to abbreviate TRT, and fracture risk in older hypogonadal men. And I'm going to talk about some planned studies that we have to address some stuff related to this. Okay, so how do I advance? There we go. So I think it's important to understand the prevalence of hypogonadism in older males. And so I'm going to show you two sets of data from our lab. One is a clinical trial in which we enrolled 203 men, all veterans, all over the age of 60, and we assessed their testosterone levels. And on average, they were about 400 nanograms per deciliter. And you'll see that dashed red line there on the top left graph, which is about 300 nanograms per deciliter. And this is where we consider any value below that as low. So on average, men who are over the age of 60 do not have low testosterone. But if you look at the prevalence of low testosterone in this cohort, it was about 1 in 3 men, which is a lot higher than what you see in the literature. That suggests either this veteran cohort is different or perhaps the literature is wrong. So we wanted to look at this. And so we used NHANES data, which, if you're not familiar, is population representative data from the US population. Oh, yep, thanks.
Thank you. You click there and swap presenter.
Okay, better. And so in our NHANES data, we found 2,400 men who had testosterone levels, and we categorized them into young men in black, middle-aged men in gray, and what I'm going to point out is the older men here in yellow. And if we look at the average testosterone of the old men over the age of 60, what you'll see is it was very similar, about 400 to the veteran cohort we saw. And if we look at the prevalence of low testosterone, you'll see it's near identical in the NHANES data and our veteran cohort. So these are both population representative samples. So we actually think that the prevalence of low testosterone in older men is quite a bit higher than what you traditionally see in the literature, which for this population would be around 10 or 15%. And the rationale or the reason for that is that in a lot of these studies that have looked at prevalence of hypogonadism in older men, they've excluded a lot of conditions like older men with cardiovascular disease and obese older men, and a lot of other conditions that, and these two studies did not exclude those. So this is true population representative data. So when we got interested in bone many, many years ago, my research spans the preclinical, clinical trial database spectrum. So this is the first study we ever published on bone about 20 years ago. And this is an animal study, males at the top, females at the bottom, and this is a histologic section of the proximal tibia. And so what you'll see here is this purple stain is the growth plate at the proximal tibia, and we're interested in quantifying the trabecular bone, which is stained black below the level of the growth plate. And so these are just normal intact animals. And in this study, we gonadectomized these animals. So it's an orchiectomy in females or an oviectomy in males. And what you see is there's a 55 to 70% loss of trabecular bone that happens within 3 weeks. And if you give back supraphysiologic doses of testosterone, in males, you fully prevent this loss, and in females, you partially prevent the loss of trabecular bone. And so the point here is that loss of sex steroid hormones drive bone loss. And if you give back testosterone in either sex, you can prevent at least part of this bone loss. We also take out the bones to test their mechanical strength, and we do so with a 3-point bending test, which is pictured there at the top. And if you assess the whole bone strength, you'll see the yellow highlighted bars there are those animals that received gonadectomy and testosterone, and they're stronger in the males on the left side and stronger in the females on the right side. So there's a histologic phenotype, and there's a whole bone outcome, which is improved bone strength. So backing up just a little bit, why do fractures matter? And this is just one article, but there's lots of them like this. And this is 12-month survival. Can I get rid of this right here? How do I get rid of hide? See if I can do it on my laptop. There we go. So this is 12-month survival following hip fracture in men on the top blue and women in the bottom pink. And this is all veterans over the age of 65, 43,000 individuals. This is not our data. This is published data from another group. And so the y-axis is odds of mortality, and the x-axis is days to mortality following hip fracture. And so this red line here is 1 year. And so the takeaway from this is that 1 in 3 elderly male veterans die within 12 months of hip fracture. And so this is why we're interested in bone, is maybe we can target this as a preventative strategy. So back to testosterone. When you administer testosterone, it is a hormone, and it binds androgen receptors that are present in bone and any other tissue, and it exerts direct biological effects through that androgen receptor. But testosterone is also a prohormone for 2 other biologically active sex steroids, dihydrotestosterone, or DHT, and estradiol. And so testosterone undergoes tissue-specific metabolism in bone and in a lot of other tissues via the 5-alpha reductase type 1, 2, or 3 enzyme, which produces DHT, or via the aromatase enzyme, which produces estradiol. And so long story short, we've done a lot of work to show that the conversion of testosterone to DHT is not necessary for the actions of testosterone to produce bone protective effects. And a lot of other groups, like Joel Finkelstein and Sandeep Khosla, have done some really intricate work showing that estradiol at least partially mediates the effects of testosterone on bone in men. So a little background here. In 2004, which is around the time that I joined the androgen field, the Institute of Medicine published a report in which the committee, or now called the National Academy of Medicine, the committee recommended short-term efficacy trials to determine if there are benefits of testosterone replacement therapy in older men. And the reason they recommended this is because at that time, 20 years ago, there was no definitive evidence that testosterone replacement therapy had any benefit in any tissue. And so they said, we want short-term efficacy trials because in the absence of efficacy, this drug shouldn't be prescribed. And so there's no reason to look at safety. So they said, do efficacy first, show that it works. If it does work, then let's look at safety. So there was many trials published since then, and the Endocrine Society came out in 2010 and published guidelines on testosterone replacement therapy. And at the time, they said that there's an increase in lean body mass of about 2.5 kilograms in men receiving TRT, and a reduction in fat mass of about 2 kilograms. And so this is no change in body mass. But they said that there's inconsistent evidence whether TRT improves muscle strength, physical function, or bone mineral density. And in our lab, this is not what we see. We have very consistent evidence in our lab. And so we wanted to approach this question, and we did so via meta-analysis. And so if you're not familiar with this, the red dashed line is the line of no effect. Any value that falls on the right side favors testosterone, improving lean body mass. Any value that falls on the left side of that dotted line favors placebo. And so we took all double-blind, randomized, placebo-controlled trials that administered testosterone either via transdermal or intramuscular routes, which are the 2 most commonly prescribed routes. And we looked at the overall improvement in lean body mass, and it was about 3.5%, which is right in line with the Endocrine Society guidelines. But we went a step further, and we divided these into studies that administered transdermal in yellow or intramuscular testosterone in green. And we saw a difference. Transdermal, on average, improved lean body mass by about 2%, and intramuscular improved it by about 6%. So 3 times more benefit from intramuscular testosterone versus transdermal. That was lean body mass. which is not skeletal muscle mass. So if you believe lean body mass is skeletal muscle mass, you should be able to detect a functional outcome from this. So we looked at this in terms of lower body strength, same approach. The overall effect was about a 5% increase in lower body strength, not that impressive. When we divided this, the effect was entirely driven by intermuscular testosterone studies, which showed about a 10.5% increase in lower body strength, and transdermal resulted in, on average, no improvement in lower body strength. So this is pretty impressive considering there's no exercise interventions in these studies. This is a pure drug effect. And I did want to point out one study, the TEAM study, which I will talk about in a few slides, which was a transdermal study that gave a low dose or a high dose of testosterone. It was conducted here at Colorado. So I said that our data does not suggest there's inconsistent evidence. So this is one clinical trial conducted in our lab in which we enrolled 60 men all over the age of 60 who had low total or bioavailable testosterone. It was 12-month double-blind randomized placebo-controlled trial. Factorial design in which individuals received testosterone alone, finasteride alone, which is a type 2 5-alpha reductase inhibitor, the combination of those two, or a double placebo. And I'm just going to show you bone mineral density data at the lumbar spine. And so in red, that's testosterone alone. In green, that's testosterone with finasteride. And you see that they're identical improvements with these two drugs. And it was a 4% increase in lumbar spine bone mineral density, which we considered quite impressive because this is online or even slightly greater than the effect you would get from a frontline bisphosphonate given to older men. So we thought maybe there is a difference in terms of how you give testosterone for bone, much like for muscle that I just showed you. So this is very preliminary data from a meta-analysis that is ongoing, in which we looked at transdermal testosterone versus placebo in black and intermuscular testosterone versus placebo in red. And regardless of the site that we looked at, lumbar spine, femoral neck, or total hip, and this is all double-blind randomized placebo-controlled trials, the transdermal resulted in a 0.5% to 1% increase in lumbar spine or any of those site bone mineral density. And the intermuscular resulted in a 2 to 4% increase, which is, again, right in line with our clinical trial data. And so it's a 3 to 4 times greater BMD increase in response to intramuscular versus transdermal, suggesting, again, that perhaps how you give a drug matters. So the Endocrine Society updated their guidelines in 2018. So we've been talking about bone mineral density, and bone mineral density is a surrogate for fracture, but it is not predictive necessarily of fracture. So in 2018, the Endocrine Society said testosterone is not approved for treatment of osteoporosis or for reducing fracture risk in men. They also said T may reduce fall propensity and fracture risk. However, there are no randomized trials that assess the effect of T on falls or on fracture prevention. So I want to introduce you to a study, the TRAVERSE study. This is not from my lab. So if you're not familiar with the TRAVERSE study, this is 5,200 men, all between the ages of 40 and 80. This is a prospective randomized controlled trial. All these men had low testosterone and pre-existing cardiovascular disease or elevated cardiovascular risk. They were randomized 1-to-1 to receive transdermal testosterone versus placebo, and they were followed for 3 years. The primary outcome of this study was major adverse cardiovascular events. That's not what I'm showing you here. The take home from the MACE study was there's no difference in major adverse cardiovascular events between testosterone receiving men and men who received placebo. Here, they looked at a secondary outcome, which was fracture risk. And so I want to take a quick poll of the audience. Based on what I've shown you so far in our animal models, testosterone increases bone mineral density and it increases whole bone strength. And in men, testosterone increases bone mineral density. What do you think the results of this study were? So I'll give you 2 options. One option, testosterone reduced fracture risk. Raise your hand if you think that's the outcome. Okay, half the audience. The other option is there was no difference between fracture risks and those receiving testosterone. It's about the other half of the audience. It was a trick question. Surprisingly, they showed that regardless of how they looked at fractures, those men receiving testosterone had numerically higher fracture incidence than those receiving placebo. And so this is whether you look at non-high impact clinical fractures, whether you look at all clinical fractures, or whether you look at clinical fractures in those not taking osteoporosis medication. And so their conclusion among middle-aged and older men with hypogonadism, fracture incidence was numerically higher in men who received testosterone than those who received placebo. And if you look at any of these graphs, the separation between orange, which is receiving testosterone, and blue, which is receiving placebo, happened very early, and it was continuous. So this is concerning and hard to understand. So if we believe that that's true, then perhaps if we're prescribing testosterone, we should be prescribing another therapeutic approach along with testosterone to prevent this. And so the TEAM study, which I mentioned earlier, was a study in which it was conducted here. You'll notice a lot of the authors' names you probably recognize, including the first author and the middle authors and the last authors, because they're all part of this division. And this study gave testosterone alone, progressive resistance training alone, the combination, or neither of those. And so I've highlighted in yellow several of the primary outcomes. In top is total fat-free mass. In the middle is appendicular fat-free mass. At the bottom is fat mass. And in yellow, what you'll see there is that's the combination group. The light gray bars are the 0 to 6 month change. The dark gray bars are the 0 to 12 month change. And the concept that I wanted to point out here is that the combination of these was better than any of the individual treatments. So the TEAM study collected lumbar spine and hip bone mineral density, and this data is available, but they didn't publish this data yet. And so the questions that we're interested in my lab are testosterone replacement therapy administration routes equivalent? And we're addressing this by completing that meta-analysis that I talked about early, which is comparing bone mineral density changes in men who received either intramuscular or transdermal testosterone versus placebo in randomized clinical trials. We're also interested in the question, does testosterone impact fracture risk? We are planning to approach this from a retrospective analysis perspective to look through VA electronic medical records to evaluate fracture rates in older veterans who either received testosterone or did not after a diagnosis of low testosterone. And we're also interested in determining, do testosterone replacement therapy and progressive resistance training produce additive bone mineral density gain? And we're assessing this through the TEAM study data. So the questions that I want to pose to this audience are, from a clinical perspective, what do you think should be controlled for in these projects? Conditions, contraindicated drugs, previous fracture history, et cetera. As clinicians, what data would help you make informed decisions about the appropriateness and/or risks of testosterone in relation to bone mineral density specifically? And is anybody interested in joining these projects? And so we're interested in fellows, students, others, anybody who wants to provide clinical input, specifically anybody with meta-analysis or database perspectives, because these are largely outside of my wheelhouse. And so I wanted to thank the people who are already engaged in these projects. And I'm going to back up just one slide. Whoop, wrong way. And ask if anybody has questions or comments specifically related to these or other issues.
I'm going to apologize as a facilitator and thank the audience for letting us run a little bit over time as we work out the kinks for this format. If you could please fill out the feedback forms and also just continue these conversations. At the very least, I hope this is an opportunity to see some of the research that is going on in our division in a quick format, and also think a little bit more about how we can collaborate. All right, take care.